In any case, like where I'm going with this is that like George's lab was this place where people were working on super ambitious things. And, these days, and when I was there, the big thing was CRISPR. Like CRISPR-Cas9 was this monumental change in what we could start to imagine as possible with biology. Obviously, the story of CRISPR-Cas9 is not one that originated in George's lab. Sort of leave the debate there.
We don't have 8 hours.
Yeah, we won't do it. But, Jennifer Doudna's like, at Berkeley, they were the ones who were like showing there's a system, it exists in bacteria, and actually, like maybe we can do something interesting with this. And then George and Feng at the Broad Institute, so Harvard and MIT, showed actually we can apply this system to mammalian cells. Turns out we can edit human cells. And it's like something people have talked about. Like gene therapy has been this idea for, probably since the 80s, 90s. When we started to understand genetics, we started to understand that there are certain diseases that are driven by aberrant mutations in our DNA. Like the code that makes us run can sometimes come messed up, or it can become messed up over time. And with CRISPR-Cas9, all of a sudden, like the idea of what we could do with that was very different. Like, before that, you had a disease, maybe you had cancer, maybe you had something else, and you try to find a small molecule. And a small molecule, you don't really know. Maybe sometimes you don't really know exactly what it does, but it improves the condition or it doesn't improve the condition. But with CRISPR-Cas9, all of a sudden the idea is like somebody has a disease and we can figure out exactly what is causing that disease. So maybe it's a specific mutation in the DNA, and this is making like a broken protein. And that broken protein is making somebody very sick. And, we can actually just reverse that. We can just fix it. And now all of a sudden, the pitch was like a one-time thing.
Right. Yeah. Instead of perpetually treating it through a drug, you just get rid of it.
Yeah, exactly. That's been the idea. I think there's some smoothing out that needs to happen, but that was really the idea. And so I've been working around that idea for a long time. I really bought into this concept of like, can we give people like agency over who they are, like their DNA, right? It drives so much of what it means to be, Ashlee or Aaron. There are things that are in our DNA that are written there, and it kind of dictates a lot about us. And unfortunately, for some people, it means that they have to deal with pretty serious conditions from birth, like and onwards. And so CRISPR-Cas9 has had this huge, there was the original thing, but then all of a sudden, people started to work on like better ways of using the system. And we'll talk about, David Liu has been a big part of the KJ story. But, if I had to sort of summarize the whole thing, it's like CRISPR-Cas9 all of a sudden allowed us to really imagine just going way upstream, like what causes the disease. Let's just go all the way up to what started it and just fix it. And now you're done.